Good afternoon, everyone. I hope you all are having a wonderful day and a great week. And welcome to our lecture for Microbiology for Health Sciences. And today we are going to be covering Chapter 7 from our textbook. And this chapter includes information about viruses and prions. To begin, we're going to start discussing viruses. Now, first and foremost, viruses are not cells, but they are a unique group of biological entities and they can infect every type of cell. Now, viruses can be classified as active or inactive. Active viruses are viruses that are inside the cell and they're only inside the cell. Inactive viruses are macromolecules that are outside the cell, thus they cannot function. Viruses can be counted into total numbers inside the body, and those are called a virome. An example of a virus here would be a Epstein-Barr virus. And the Epstein-Barr virus is a type of herpes virus, and almost everybody is infected by herpes virus at least once in their lifetime, whether that be in the genitalia or on their mouth. Now, this image here to the right is just a little image of COVID under the microscope, just to give an example of what a virus may look like. Next, we're going to talk about the size range. Now, the size range of viruses is that they can come in many different shapes and sizes. But they're so small that they need a microscope to be seen. The range of sizes can be from the smallest of parvoviruses, which can be zero point 0.2 microns in diameter, to the largest of viruses, which can be larger than some bacterias, which ranges from zero point 0.4 microns to 1 micron in length. Now, over here to the right, we have a little representation of a yeast cell and E. coli, and then other different types of bacterial cells, along with the sizes of viral cells. As you can see here, the yeast cell is much larger than all of the viral cells, but then there's some viral cells that are pretty big within themselves. Now we are going to talk about the viral components of a virus molecule. And viruses can contain either DNA or RNA, but they can't contain both at the same time. Now, the RNA can sometimes come in one or two enzymes, and the DNA is linear and it in double strands and linear, or it can sometimes be double strands in circles. The RNA can be double stranded or single stranded, but mostly it's going to be single stranded. And viruses also contain a protein capsid, which is inside and which is in the virus and houses most of where in your DNA and different proteins are going to be. Now, viruses can be either naked or enveloped. A naked virus is a virus that includes only the nucleocapsid, and the nucleocapsid of naked viruses are said to be rigid and they're tightly wound into a cylinder-shaped package. Now, enveloped viruses they have a helical nucleocapsid, and these viruses are more flexible and they tend to be arranged around a looser helix within the envelope. And to the right here, we have a representation of a naked virus. And as you can see, it's not enveloped, but it's more rigid with these spikes that go around it. And here we have a enveloped virus. As you can see, the yellow part here is the envelope that goes around, which can cause it to be more flexible. Now we're going to move on to how viruses are classified. Classifying viruses can be very difficult because they're so small and there's so many of them. Um, They're usually classified based on their structure, the chemical composition, and genetic makeup, which is fairly similar to how different types of microbes are classified as well. There are 59 orders of, sorry, sorry. There are 59 different orders of viruses, as well as in these orders, there is 189 different families. Now, the orders, when you write them out, they're written with virales at the end, and then families, when written out, are written with viridae at the end. Now, over here to the right, we have a little chart that gives us some examples of classifications. And like I said just a few seconds ago, in the orders, they have virales at the end, such as herpes virale. And then the families will have viridae, such as herpes viridae. I apologize if I'm mispronouncing these. Um, Now we're going to move on to our next section, which is the multiplication. And the multiplication process of viruses goes through several different phases, and these phases are absorption, penetration, uncoating, synthesis, assembly, and release. The length of these different phases of this cycle can vary, which in polioviruses, it can take up to 8 hours, and then in herpes viruses can take up to 72. And when we get to the release stage in the multiplication phases, in non-enveloped and as well as complex viruses, the cell lyses, which means when it reaches maturation that the virus is violently protruded from the cell, or it ruptures, basically leaving the cell pretty pretty messed up in the cytoplasm, and this occurs in the cytoplasm and the cell nucleus. And in enveloped cells, no, I'm sorry, enveloped viruses, they're released through budding, or also known as exocytosis. And this happens in the membranes of the cytoplasm, the nucleus, the endoplasmic reticulum, and the different vesicles that occur. And viruses with induced damage are known as cytopath is known as cytopathic effects, or CPEs, and this leaves the cell altered with a chain which can change their positional orientation, or changes their shape and size, and also they're inside the cell. It can change structures in there as well. Now we're going to go on to talking about viruses that infect different types of bacteria, and these are known as bacteriophages. Most bacteriophages have double-stranded DNA, but it can also exist as single-stranded DNA, and as well as a types that have RNA. And these can be classified as T-even, and T-even bacteriophages go through similar stages of animal viruses when they absorb to hosts, and they use specific receptors that are on the bacterial cell to attach. The most common types of T-evens are T2 and T4. And bacteriophage infections can also cause, and will more than likely always cause, the lysis of the cell. And this happens because the cell has said to be in the lytic phase of the cycle, and this usually damages the cell, as we discussed earlier. Viruses that infect other viruses are called virophages. And to the right here, we have a chart that lists the differences between the different types of multiplication phases between bacteriophages and animal viruses. We can go through one of these, and which we're going to go through absorption. And in absorption of the bacteriophage, there is a precise attachment of the speed tail fibers, and these attach to the cell wall. And then, as we discussed earlier, in animal viruses, they attach to the capsid or the envelope of the cell's receptors that are on the surface. Now we're going to talk about different types of cultivating techniques. And before we get into that, the cultivating techniques that are in vivo use living embryos, and then the cultivating techniques of in vitro are use cells or tissues that are cultivated in the labs. When they do these cultivating techniques, they use the most commonly used species are chicken eggs, duck eggs, and turkey eggs. And when they do these, they usually drill a hole through the shell, which makes a small window to where they can inject the virus through the shell and into the embryo. And the most common types of cultures are the animal cells, and they um, are commonly grew in sterile dishes or sterile bottles, and these dishes and bottles are under special conditions such as environmental growing conditions and so forth. Now we're going to talk about how these viruses affect the human health. And as stated in the first bullet, it's impossible to know how many actual viruses there are in the world, and it's impossible to accurately measure what parts of the world they originate in or where they're placed. And there's 268 viruses that are known today. And although still think there's a lot more, because it's impossible to measure. And as of 4 years ago, we got COVID and the widespread effects of that, which led to discoveries of the different types of COVIDs and so forth. And which also, it was a good thing for scientists because it gives them more knowledge of viruses and how different viruses are affected and how they affect humans themselves. Not all viruses, though, can result in death, but some do, and some have extremely high mortality rates, such as rabies, which is usually caught from different animals, such as raccoons, and then sometimes dogs and wolves. And then Ebola, and Ebola was a big epidemic back in the mid and early 2010s, and it gave us a lot of knowledge on different types of viruses as well. Inferions, which are inside humans, can also help prevent viral infections. They help fight viral infections, and they also combat different types of cancer as well. And some viruses can lead to cancers, and they can lead to cancers such as liver cancer and cervical cancer. Now, last but not least, we're going to talk about prions. Prions, they're similar, but they're different from viruses in their size and shape, as because prions are smaller and they usually take unusual types of forms. And we can see that right here to the right here. This is a microscopic image of a prion. If you look at this one under my mouse, you can see that it's kind of looped in different phases and so forth. And prions, they're primarily composed of proteins, and they can be labeled as prions can be labeled as chronic, and they're very prominent in animals and as well as humans. And a one type of prion that was covered in, or that we cover in our textbook, is the Creutzfeldt-Jacobs disease, or CDJ, and this causes degeneration and death. And this prion will degenerate the nervous system, which can lead to impaired senses, dementia, and as well as unwanted muscle contractions. Prions in animals can include things such as mad cow disease, which was very prominent in the 80s and 90s. And it was found that it infected the beef of these animals and it degenerated their brains. And when people unknowingly would eat the beef from these animals, it would also infect the humans as well, which would cause degeneration of the brain and would lead to death. There is also a type of prion that affects deer if you're a hunter, and it has the same effects of mad cow disease essentially, and will leave the deer disoriented and out acting out in different rationalities, and which until it ultimately just dies. And this concludes our lesson for today. I hope you all have a great, wonderful rest of your week. And just remember to study hard and to keep up on all the information in the books, and you will be fine. Thank you.